Mitochondria rely heavily on the nuclear genome, despite having their own DNA. Thus, a highly regulated mechanism of import is required. This method helps study the process and how it can adapt to external stimuli. This is the only biochemical technique available to quantitatively evaluate protein import into the various subcompartments of the mitochondria. The viability of mitochondria is implicated in various disease states, so understanding how protein import is regulated may help contribute to novel therapeutic approaches that target mitochondria.
Extra care is required during the isolation to ensure the viability and integrity of the mitochondria. Thorough mincing of the tissue and gently resuspending all subsequent pellets is necessary.
Demonstrating the procedure will be Ashley Oliveira, a PhD student in my laboratory.
To begin isolation of mitochondria from skeletal muscle, remove fat and connective tissue from the muscles and mince the tissues on a pre-chilled watch glass until it is a homogeneous slurry. Place the minced tissue in a pre-chilled 50 milliliter plastic centrifugation tube and record the exact weight. Then, dilute the minced tissue tenfold with buffer 1 containing ATP. Use an 8 mm twin blade homogenizer to homogenize the muscle sample at a power output of 9.8 Hz for 10 seconds, ensuring no visible chunks of muscle remaining. After spinning the sample at 9,000 times G for 10 minutes at 4 degrees Celsius, resuspend the pellet carefully in roughly 95 microliters of the